and welcome to a new episode of the Computomics podcast. My guest today has received multiple awards and recognition, including one as one of 25 Germans who will shape the next 25 years. He's the scientific coordinator of the Marie Curie Network and currently the deputy head of the Department of Biosystem Science and Engineering at ETH Zurich. Welcome to the Computomics podcast, Karsten Borgwardt.
Thank you very much. It's a pleasure to be here.
Pleasure to have you. Karsten, we agreed we'll use first name as per usual. Karsten, what is your current focus area at ETH Zurich? What's your research focus?
So I'm the professor of machine learning in the Department of Biosystem Science and Engineering, as you said. So it's a mixture of machine learning and the life sciences. That's actually my focus, developing machine learning data science techniques for solving problems in the life science domain. So reaching from plant biology to medicine, we are developing algorithms for answering questions in these biomedical research areas.
And knowing not too much detail about your work, could you give an example of such a problem that you tackle?
The one very important problem for us at the moment is, for example, in the medical domain, where we are trying to use all the data that is being collected about a patient in the intensive care unit to predict whether complications will arise during the further stay in the intensive care unit. For instance, whether this patient will develop sepsis. So that's extremely hard to predict. So sepsis is also more informally known as blood poisoning. It's extremely hard to predict whether someone will develop sepsis during an ICU stay. And we want to support this recognition problem by using machine learning. So we want to build algorithms that can, from all the data that's being recorded, from all the physiological data that's being recorded about the patient, from all the lab measurements, lab tests that are being performed on samples from the patient, using all of this data, we want to make an accurate prediction whether this person might develop sepsis in the next few hours. Mm-hmm. This is like one typical example of where we use machine learning. And in another recent study, we used machine learning to rapidly predict whether there's antibiotic resistance in a given patient. So given that a patient has been infected by a bacterium, for example, we predict whether there are antimicrobial resistances that should be respected when choosing a treatment for this patient. So these are two examples from the medical domain. In the biological domain, we also look into more basic research questions like which genes or which variation in which genes in an organism are associated with phenotypic changes. So like a classic question from genetics, which genome sequence variants are associated with an observable change in the phenotype, so in characteristics of an individual. And there we are developing novel algorithms for this task. For instance, algorithms that take not one gene or one sequence position into account, but multiple or even interactions between multiple genes or multiple genome positions.
Those were quite a few examples and also quite a broad spectrum that you're dealing with. If I were to pick just your second example of antibacterial resistance, rapid recognition of that, how can I envision you using machine learning for that purpose? How would that work?
So how we did it in this recent study, this is a work that appeared in Nature Medicine in 2 weeks ago, in January 2022. So in this work, we used hundreds of thousands of so-called mass spectra measurements, multi-TOF mass spectra measurements that had been collected in laboratories all over northwestern Switzerland. And you can think of these as fingerprints of proteins in a sample from a patient. And we use these profiles to train a machine learning algorithm to distinguish between such profiles that had been collected from a resistant sample from those that had been collected from a sensitive sample. So we gave a lot of examples to the machine of these protein fingerprints, you could say, such that the machine learns to distinguish between those that represent a resistant example from those that represent a sensitive example. And the key to making this work was the access to a very big collection of data. So we combined data from the University Hospital Basel with data from various other laboratories, as I said, from northwestern Switzerland, from other hospitals in this area. And this allowed us to have a collection of hundreds of thousands of these protein fingerprints of multiphoton mass spectra, so more than 300,000. And we had more than 700,000 associated resistance phenotypes. So where someone where someone had experimentally examined whether this sample, this pathogen, is resistant to a particular antibiotic or antimicrobial or not. So this huge collection of mass spectra plus associated resistance phenotype allowed us to then use algorithms that can, from such massive collections of data, learn the connection, the correlation in most cases between mass spectra properties and the resistance phenotype of interest. And then when presented with a new mass spectrum from a new pathogen or from a new infection, a new patient in the hospital, then we can make a prediction how likely it is that there's a certain resistance existent in this particular case.
And then accordingly change treatment if necessary, if
exactly.
the resistance is likely.
Exactly. So, and this we explored together with our clinical colleagues. So one specialist for infectious diseases did a retrospective analysis using our tool. So this specialist looked at 63 cases retrospectively that had been treated in the hospital with antibiotics, and he simulated the scenario. What would he have done, or would he have changed the treatment if our prediction had been available to him at the time he initially chose the treatment? And he looked at 63 patients, whether our prediction would have made a difference in the treatment. Because
would it?
So it turned out in 9 cases, yes, 9 out of 63, yes. And in 8 cases, this would have been beneficial. So we would have de-escalated the antibiotic treatment, used a more specific antibiotic in 8 cases. And in the 9th one, we would have recommended an unnecessary escalation of the antibiotic treatment. So in close to 90% of the cases where the algorithm triggered a change in the treatment, it was beneficial for the treatment. That's the first hint that this could actually really have a clinical impact, our predictions, and that it's not just a theoretical exercise to develop such an algorithm.
Not that we want to judge theoretical exercises, but
especially I'm a theoretician myself, so I have no reservations here. Yeah.
Of course, but again, with being able to apply it, obviously that's another step and a very valuable step.
Yeah.
You also referenced the sepsis as an example, and I know that you actually lead the what's called the personalized Swiss sepsis study. Could you go into a little more detail? And I'm especially interested in the personalized part of that name of the equation.
Yeah, so let's go into the details of this personalized Swiss sepsis study. So it's called personalized because we try to make predictions for an individual patient based on the properties of that patient. And so far, in phase 1 of this personalized Swiss sepsis study, this is the electronic health record of that person. So all physiological measurements of that person in the intensive care unit go into our data sets. So, for example, heart rate, blood pressure, respiratory rate would be typical examples here, like lab tests, for example, on blood samples from that patient, or other information about the health history of that person, like are there any comorbidities, for example. So that's the type of information that we are currently collecting for the study on each individual patient. And this data is then being used to train a machine learning model to then be able to make for a specific individual patient a personalized prediction. That's why we speak of personalized medicine here. And it's called Swiss Sepsis Study because we are harmonizing the data from all Swiss university hospitals from their intensive care units for this purpose. So this is
from all of them? From
all 5, yeah. So Lausanne, Geneva, Bern, Zurich, and Basel. They all come together and we harmonize their data into one joint data set, into the same format, such that we can learn a model across these different sites and also evaluate our model on these different sites and then make predictions of whether sepsis might happen or not. So these are the two aspects, so like really a nationwide effort to collect data for this task and together build a machine learning model for accurately detecting and predicting sepsis.
Very interesting. And how long is the study? Because it's still running at the moment, right? How long will the study go?
So we started in 2018, and it's a long-term effort. So you can probably imagine that even getting 5 hospitals or nationwide to agree legally to exchange the data and to harmonize it in one way or in one central location was a big legal effort, in fact, to organize this properly. And now afterwards there was a huge data engineering and data curation effort to harmonize these different data. So Switzerland has 4 national languages. I mean, this alone causes some challenges here, but beyond that, so really ensuring that variables that we get from different hospitals have the same meaning, that the measurements are comparable, that's a huge effort of data curation. And we are now completing this effort, and we are now in these months, in these weeks, reaching the point where we can really then do machine learning on this data and do predictions. So now we have in the order of 17,000 patients in this harmonized data set, and it's constantly growing. It's roughly growing by 800 patients a month. So for more than 2 years now, we have been including, or roughly 2 years, we have been including patients into this joint data set, and it's constantly growing. And this is what I now describe as phase 1 of the project. We also are very eager to start phase 2. There we will also collect molecular data about these patients, like different omics levels that describe the patients that develop sepsis during their stay in the ICU.
And so we're now kind of heading towards the end of phase 1, I take it, and we're about to enter phase 2 where you take molecular data. And how long is that set to go, or when do you expect to also to have that first machine learning phase completed, and maybe even have the first results, kind of intermediate results to go off of? So
for the phase 1, I think we'll have the results in the next 6 months, so maybe in the first half of 2022. And for the molecular one, of course, depends also a bit on how much COVID is keeping the ICUs busy and whether there's enough time to pursue this project. I believe this, if let's say the pandemic, or let's assume the pandemic now ends, let's be optimistic, then fingers crossed, then maybe 2 years down, we'll need to collect as a data set of sufficient size, and then we can do the machine learning analysis. So I expect that this will be something for first half of 2024, 2 years from now.
Yeah, those are quite long time frames. And also I thought it was really interesting, that aspect of really to have that, to train that machine learning algorithm, you really need good data. You need enough data and you need good data, and the challenge that poses. What do you think will be the role of these of national or even European or international projects for these types of machine learning projects that you're doing?
Yeah, so we are very quickly in machine learning reaching the point where in many research questions, a single hospital is not collecting enough examples to really do a form of big data analysis. So maybe even the antimicrobial resistance prediction project I mentioned before is the counterexample. So there, the University Hospital Basel alone collects like tens of thousands of these measurements, these multi-top measurements in clinical routine. So that's maybe the counterexample. There are such applications. But then sepsis, if you want to study sepsis at the molecular level, then it's known that sepsis is very heterogeneous as a syndrome. So it may be caused by different pathogens. So if you want to stratify for a particular pathogen, for example, that causes sepsis, then your case numbers drop so much, then not only is then like one hospital too little to study this, maybe even one country, even a medium-sized country is then too small to study an important syndrome or condition like sepsis. So we really depend on large data sets, and that means we have to work at an international level. We have to bring together data from different sites here to really reach the the orders of magnitude in our data sets that allow us to do what one could call like big data analysis. And so that's one big point. So we need this collaboration to have sufficiently large data sets. The other point is, of course, and that's also fundamental in medicine, but in all areas of machine learning in the life sciences, we have to validate the predictions that we make externally. So if sepsis prediction works in Basel, this is no guarantee that it will work in Zurich. So maybe there is an aspect of the Basel data that gives away that someone will develop sepsis. So there may be a form of confounder that the machine learning will pick up, and this confounder may not be present in Zurich. So then you shouldn't like celebrate if the algorithm works in Basel. You should then take it to other locations, slightly different data, see whether it generalizes to this other setting, see whether it is robust in this other setting and can still make good predictions. And this we did, for example, in the antimicrobial resistance prediction. So we had their data from these different hospitals and laboratories in northwestern Switzerland, and we tested whether when training on one side, on data from one side, we could then generalize the predictions to other sites. And what we saw is that we could largely do is there was some like drop in performance, but not a drastic one, not in the sense that it got random on the other side. So there's some effect of location, but it's still working in most cases. Yeah.
Mm-hmm. So that would always have to be, that's how I take you now, the last step would have to be this kind of checks and balances of, okay, now we need to validate, now we need to go somewhere else, have a diverse data set,
exactly,
and test if it works the same way, if it spits out, exactly. if you pardon the general term, spits out.
That's extremely important in machine learning and medicine, this mm-hmm. point. It's maybe less dramatic when you do machine learning for basic research in biology. When you grow plants in a greenhouse under controlled conditions like Arabidopsis. So in earlier phases of my career, I also worked on Arabidopsis. So this is when you look at like laboratory strains of Arabidopsis, the environment is more controlled. Of course, like a human patient is completely different. It's a completely different scenario. So in model organisms, you can control the environment in the lab much more. So there you can generalize more. But in a setting where the environment is so variable, as in a hospital or the living circumstances of a living environment of a patient, there you really have to take this to a different location and see whether the predictions that you made in the first location also generalize to the second location, mm-hmm. because you didn't have the chance to control the environment in the same way as you could do with model organisms in the lab.
How do you see the outlook for machine learning, given that it is, you mentioned it a bit earlier, quite the challenge to bring together so many different players from either different regions, different nations? You know, there might be quite a lot of red tape to deal with. How do you see this working in the years to come?
I think it's a huge enterprise, a huge undertaking to make machine learning in medicine work. It's not like a small research project that maybe one professorship can do on its own with the team, with a small team. It's really like bringing together different hospitals, the levels of expertise, or different points of view, like the clinical one, the machine learning one, the legal one, the ethical one, the views or the perspective of the patients. So there are many parts of this system that you have to get to work, so that you act legally and ethically in your research, that you do machine learning research that has a clinical impact. So why would we do all this effort if then we just have some like models that would never really help the treatment in the clinic? So you have to be in a steady exchange with the clinicians and also learn about their perspective on what is useful and what is not useful. And maybe they learn about our perspective, what is doable and what is not doable with algorithms. So a very good collaboration with clinical researchers is key here, and managing these various different and challenging aspects from legal clarifications, ethical approval, IT requirements, data harmonization. Mm-hmm. So there's a whole workflow of things that have to be taken care of. So it's a massive enterprise, as I said, and you have to have experts for all of these various domains. So maybe we even have to do more division of labor than we do so far. So we need like bigger teams where there are legal experts, where there are experts for the ethics approval, experts for IT issues and data harmonization and so on. And I think also we are going in this way, and Switzerland is doing very well. Is pushing in exactly this direction, that we have a national infrastructure for doing such projects. I think that's a very good direction, I believe.
Yeah, for sure. That was while you were saying we need bigger teams, maybe more specialization. I was also thinking also templates. Ideally, you learn just like the machine learning algorithm learns with the data input. The team or any such structure combination of people working together would hopefully learn from previous projects and maybe even share. I think that would be a very interesting aspect, to have this open science aspect to the process of how did we make this work? What were the challenges? How did we tackle them? And also what didn't work, and what have we learned, how we can do it better? Is there any such effort as well?
So in fact, the Sepsis study is supposed to play this role. And I think, and from my perspective, of course, I'm biased, but it is playing this role very well. So it's a so-called driver project for personalized health in Switzerland. So one of 3 driver projects where all of these challenges shall be exposed and shall be first encountered in a project of this scale. And I think I can speak for myself, I can speak for my co-coordinator, Adrian Ekli from the University Hospital Basel. So we together lead this effort. We and the team of more than 20 PIs, we have encountered all of these challenges that I previously described at these various levels now since 2018. So we can give, I think, a very complete and very long account of what the challenges are when you try to make such big machine learning medicine projects happen. And I think this has a model and a template character for future studies in the field. And so we will share this.
So it's basically 2 studies going on at the same time. You could say there's your actual research subject, and then also there will be some kind of pulling together the learnings to create a template within this driver project.
And this made this project rather unique in my career, in the sense that this was a project where I didn't only have to solve like a intellectual challenge in that sense, but I and of course the whole team, the whole consortium, we had to build an infrastructure to solve the problem. So that made this particular project different from many other smaller scale projects I've done in the past, where I had to solve like an algorithmic challenge or support a biological finding with my algorithms. Here it was like building a national infrastructure and then tackling the research question.
The listeners can't see you, unlike me, and your eyes like literally lit up as you said that. I can see that you enjoy that challenge of doing both, even though I'm sure it was hard.
It's good when you look back at it and know that you managed to overcome the challenges. But I can tell you, when the challenges are in the future, it's sometimes maybe my eyes are not shining in that moment. So it's good to talk about challenges that were overcome or that were solved in the past. Yes. So then,
For sure. You have, I think you've just started a sabbatical too, right?
Yeah. So my first sabbatical at ETH, since I joined ETH in 2014. Yeah. So my research will continue as usual, but for 1 semester I will not be teaching. So I usually teach a data mining course at ETH every semester, data mining 1 and 2, and now in this spring I won't be teaching data mining 2 for 1 semester.
So you will focus mainly on that sepsis study and the other research.
So this research part will continue. I don't have teaching duties for 1 semester. That's the change. Yeah.
Sounds very exciting. I have just one more topic I'd like to broach. Namely, you are also a member of the scientific advisory board at Computomics. So, if we can't not talk about it in the Computomics podcast, what does that role entail of scientific advisor on that board?
So it's a very exciting role. So I'm also a co-founder of Computomics. So in 2012, together with with colleagues from Tübingen and with the current CEO, and we founded Computomics, and I've accompanied the development of the company over the years. So these days I work in a branch of machine learning in the life sciences that is a bit further from Computomics. But back in the days in Tübingen, I did a lot of work in plant genetics, which is very close to what Computomics is working on. I think both my current research and the fields that Computomics are active in are examples for how exciting this field of machine learning in the life sciences is. Also, now that Computomics is turning 10 years this year, I think we have witnessed an enormous growth in topics and possibilities in this field. So the field has really very much grown in importance over the last 10 years. And I can see this from the things that Computomics is doing, the projects that Computomics is doing. I can see it in my own research, like how the possibilities and how the prospects and so on of machine learning in the life sciences has grown. Yeah.
And now that your role is, from originally being a co-founder to now being in the advisory board, what do you get to do still, like with that, one of your babies, you could say, right?
Yeah, so for me, it's very interesting to follow what Computomics is doing, yield prediction, for example, for plants. Of course, there are, from a mathematical point of view, similarities to predicting a clinical complication, for example. You may be using the same algorithms for that, and maybe treatments by the doctor are modeled in the same way as treatments by the farmer, like how the farmer changed the conditions under which the crops are being grown. So there are mathematically, and it's actually also my own background, or computation is my own background. So computationally, there are common topics at an algorithmic level between these different branches of machine learning and life sciences, and therefore also like between what Computomics is doing and between what I'm doing in my current research. That's also actually an answer to a previous question that you asked, about like the diversity of topics in my lab. Mm-hmm. This diversity stems from the fact that in these various branches of machine learning in the life sciences, there are fundamental algorithmic problems that tend to reoccur all over the time, like how to best model the environment, how to detect complicated, not simple, not linear interactions between genes or components of a system, if I phrase it even more abstract. So these are reoccurring algorithmic questions in different branches of the life sciences. And the diversity of my own lab stems from the fact that we are interested in solving these fundamental questions. And then sometimes when we have solved it for one application, we can also then take this knowledge and solve a different application in the life sciences. So this is also the reason why I, in my lab, work on questions in synthetic biology and in personalized medicine, for example. So sometimes there are questions that reoccur.
Right. That makes perfect sense. And it's also a really nice way to kind of close the circle, because once again, we're at the point where we say we need more data to be able to produce a better model, right? Which is, I guess exactly. In a meta way, also what you're doing, right?
And that's a key thing, I believe, in this 10-year progress that I just referred to. So over the last decade, the number of domains in which big data is available has just exploded. So these were very limited domains in 2005 when I started my PhD, for example, and still in 2010. So it was like maybe collections of chemical compounds. It was DNA sequence databases, to some degree gene expression profiles. But so these are the classic types of big data in computational biology. But over the last 10 years, or over in the 2010s, the number of application domains where big data is available has just tremendously grown. So it's very impressive how much data is available. In particular, so in the medical domain, we have one prime example, like how much medical data is now available in digital form, and it's constantly growing. It needs some effort. I think I've described this for sepsis, to bring this into a form that researchers can really use it, but there is an enormous treasure there that can be lifted. in the medical domain. Yeah, exactly. And in the biological research, so there's basic research. There are also like the high throughput technologies have further developed, and they can measure various aspects of at the molecular level. So there's an enormous wealth there of data also. And as AlphaFold has shown, maybe this I should also highlight recently, this big data can then help to solve unsolved fundamental questions in biology, like the successful protein structure prediction by AlphaFold 2 demonstrated this very recently. This may be like another type of data I should have mentioned before. So like protein structure data was also available in 2005 already. And 15 years later, this led to this big breakthrough that we have seen now with AlphaFold 2 and its accurate protein structure predictions.
So would you say it's accurate, or it's safe to say the future for machine learning is bright, given that there is so much more data, that there are more and more model projects or like you were saying, driver projects that aim to establish templates for working with these projects? The future is bright.
Yes. It is certainly bright. In particular, machine learning in the life sciences, I think it has a very bright future. It was always a bit lacking behind machine learning for like more internet tasks. I call them informally, like speech recognition, text recognition, image annotation, and so on. Because in the life sciences, it used to be much harder to get large sample sizes. It used to be much, and often still is, much harder to get a reliable phenotype. So in these early successful examples of machine learning, for example, like playing Go, the phenotype, the outcome is very easy to define, like you win or you lose. Like phenotyping someone by telling whether that person has a disease is a much harder task. So in particular, if we start talking about psychiatric diseases, neurological diseases, and so on. So on this level, you can already tell that phenotyping, determining the outcome or the output variable that you are interested in, is much harder in the biomedical domain. So we have this difficulty, we have the smaller sample size. That's why it was always a question whether machine learning in the life sciences could also replicate the big success that we have seen in machine learning for these internet tasks. And now with AlphaFold 2 and many other examples that we have seen over the last few years, I think the answer to this is yes. And I also believe there's much more yet to come. So this, I think in the 2020s, we will see like the further rise of machine learning in the life sciences. And I'm very optimistic that we'll see further breakthroughs in this field.
I am not an expert, but I would agree from a laywoman's perspective. And I definitely hope you'll be back to talk about these breakthroughs when they happen. Be back here in the podcast to talk about them as well. Thank you so much for your time, Karsten.
You're welcome. I enjoyed this very much. Thank you.
As did I, and I hope our listeners did too. We certainly learned a lot about the personalized Sepsis study, the importance of data sets, the importance of validation, and so much more. It was really a joy to talk with you about this. And to our listeners, obviously, you can listen to the episode as often as you want to really catch all the details. It was certainly full of them. Meanwhile, until the next episode, feel free to check out our website at computomics.com. And I look forward to having you with us again. Bye.